Hey everyone, Professor Carafit here, and I wanted to share with you kind of a key to the online test. I want to make sure, hopefully what I'm sharing with you is what I intend to show you. All right. So the first question is, in his transformation experiments, what did Griffith observe? And the best answer for that one is A. And by the way, remember to watch this video only after you have taken the whole test yourself. Use this to grade yourself. Do not watch this before you take it or instead of taking it yourself. That would be silly. You want to do whatever you can to increase your retention of the information, to increase your ability to perform well on the real final. So A, mixing a heat-killed pathogenic strain of bacteria with a non-living pathogenic strain, with a living non-pathogenic genetic strain can convert some of the living cells into pathogenic form. So that was that transformation experiment we talked about. He didn't know what it was that had caused that transformation, but he knew if you figured it out, you could figure out what the molecule of inheritance was. Number two, which of the following is the first event to take place in translation? Well, the small subunit of a ribosome attaches to the 5' end of the messenger RNA. So messenger RNA binds to the small subunit, then the large subunit attaches, and then we can begin translation. Which component is not directly involved in translation? DNA. DNA is not directly involved in it. It makes RNA, messenger RNA, which is directly involved in translation. What is the function of the release factor? It binds to the stop codon, and then what it does is it then stops transcription from or translation from occurring any further. It knocks everything apart. So it binds to the A site in place of transfer RNA. What is the effect of a nonsense mutation on a gene? The best answer there is that it introduces a premature stop codon into the messenger RNA. What is the function of topoisomerase? Well, topoisomerase stabilizes the DNA during replication to keep it from breaking or twisting around. Eukaryotic telomeres replicate differently than the rest of the chromosome. What is the consequence? Well, typically the 5' end of the lagging strand is going to get shorter and shorter and shorter as you go. So we drew that in lecture a little bit. We talked about it. But if you have telomerase, the enzyme that can rebuild those ends, then your chromosomes don't shrink every time they replicate. Which enzyme connects Okazaki fragments? Well, the enzyme that connects Okazaki fragments together is ligase. Which of the enzymes synthesizes short segments of RNA? That'd be primase. It makes the primer. Which of the enzymes separates the DNA into strands during replication? That's helicase. Which of the enzymes removes RNA from the old primer and adds the DNA to the Okazaki ends? That's DNA polymerase 1. And then, of course, DNA polymerase 3 is the one that builds the long strands of DNA. Transcription in eukaryotes requires which of the following in addition to DNA or to RNA polymerase? It often requires transcription factors. And remember, those can be turned on and off through chemical signals and things. So that way you can turn transcription on and off. I should say you can turn transcription on and off, sorry. A particular triplet of bases on a DNA strand is from 5' to 3, AGT. The corresponding codon for the RNA would be what? The corresponding strand would be UCA 3' to 5'. UCA, and it's anti-parallel, so it would be 5 to 3 or 3 to 5 in this case. Alternative RNA splicing can allow the production of different proteins from the same messenger RNA, or you can mess it up and have a junk protein made this way as well, but you can get different proteins from the same messenger RNA. What is meant by the description anti-parallel regarding the strands that make up DNA? Well, one of them is 5 to 3 going this way, the other one is 5 to 3 going the other way, so therefore C for number 15. Number 16, in chromatin, the DNA is wrapped around what? The best answer there is E histones, histone proteins. That's how DNA fits into the cell. The anticodon of a particular transfer RNA is what? And the best answer here, it is complementary to the corresponding messenger RNA codon. All right, the next question is going to involve using the table here. What amino acid sequence will be generated based on the following mRNA code sequence? And I believe as long as I did it correctly, the best answer is A for number 18. You'll have to check these codons against the codon table to double check that. Which of the processes transmits and amplifies a signal after it is received by the cell? So the signal is received by the cell. How is it transmitted and amplified within the cell? The answer there is D, transduction. That's usually accomplished by sending out a phosphorylation cascade or second messengers, those things we talked about in the communication chapter. Which of the following investigators was responsible for the following discovery? In DNA from any species, the amount of adenine equals that of thymine, et cetera, et cetera. Well, that was Chargaff's rule, so that was B. 21, a frameshift mutation could result from, and there's a whole bunch of options there. The best answer there is either an insertion or a deletion of a base. A base substitution does not cause a frameshift mutation. That's when you have a T and then it switches to an A or something. That is just a simple point mutation. But if you have a deletion or insertion, those can cause frameshift mutations. What is the function of DNA polymerase 3? The best answer is to add nucleotides to the growing 3' end of a DNA strand. That's the function of 3. Now 2, we already mentioned the purpose of that. It removes the primer, replaces it with DNA. What is a word that is rarely used, if ever, in science? And if you put this on your lab report, I circled it. Proves. We never say proves in science. That's not a word we use. We have support for ideas. We can fail to support ideas. We falsify ideas. We fail to falsify ideas. We do not use proves. It's a dogmatic term that shuts off our brain and makes us just accept beliefs and ideas without questioning them, which is that's the antithesis of science. Anyway, 24. At a specific area of the chromosome, the sequence of nucleotides below is present where the chain opens up to form a replication fork. So it wants to know which of the following represents the primer sequence. So remember, it's a primer, so it's going to have U's in it instead of T's. So the best answer there is A for 24. Of course, if you find one that I made a mistake on, that's fine. I filled this out as I went. I could have missed one. Let me know if you have any questions. Use the following to answer the following questions. Given the locally unwound double strand above, in which direction does RNA polymerase move? I have B, 3 to 5 going along the template strand. So it's going along the template strand in a 3' to 5' direction. Therefore, the strand that it's building is being built in a 5' to 3' direction. Mutagens are usually beneficial for an organism. Typically, no. Typically not. Mutagens can result, mutations, I should say, can result in something beneficial. But a lot of times they either have no effect or they're harmful. Which of the following is not related to the process of natural selection? The best answer there is C, the desire to become more evolved. That doesn't even really mean anything. So the desire to become more evolved is not a reason for evolution. So adaptation to the environment, absolutely. The population has heritable variation. Yeah, you have to have that for evolution to happen. More offspring are born than will survive. Yeah, that has to happen as well. Why is it important that an experiment include a control group? The best answer, without a control group, there is no basis of knowing if a result is due to the variable being tested. 29, hierarchical organization in living systems goes from lowest to highest. And which of the following statements? The best answer there is A. Just a second. Do you need something?
Yes, I want to tell you something. It's kind of important.
All right, so best answer there is A. Number 30, when two atoms share a pair of electrons, the bonding is referred to as covalent bonding. Which of the following characteristics are not necessary to being alive? So you have to go back to the first online lecture that we had, and movement is not a characteristic of life. Now, some life can move, but it is not necessary to move to be alive. 32, the partial negative charge in a molecule of water occurs because B, the electrons shared between oxygen and hydrogen atoms spend more time around the oxygen. Oxygen's more electronegative. Proteins possess all the following functions except C, they do not code for genetic information. 34, alpha helix and beta pleated sheets are examples of what level of protein structure? C, secondary structure. 35, all of the following are examples of monomer-polymer pairings except B, monosaccharides and fatty acids. Fatty acids are not part of monosaccharides, and monosaccharides are not part of fatty acids. 36, most ATP in cellular respiration is formed by oxidative phosphorylation. That is true. So if it was substrate level phosphorylation, then that would be false. Honey, I'm recording a video. I need to finish it. Please just wait. Sorry, everyone. 37, which of the following diagrams represents anaphase 1 of meiosis? The best answer there is number 1, because anaphase 1, remember, you're going to have pairs of chromosomes separating in anaphase 1. Which diagram represents metaphase 2 of meiosis or metaphase of mitosis? So metaphase 2 or metaphase of mitosis. The best answer there is what is it? There it is, 6. So we have a chromosome lined up in the middle. What is happening in 4? Let's find 4. What's happening there? Well, there we have synapsis. So crossing over is occurring. Chromosomes are paired up. Examine the karyotype below. What is the gender of this person? Well, they have 2 big X chromosomes. So this is a female. The karyotype is normal or abnormal. Well, abnormal because there is an extra chromosome at number 21. They have trisomy 21. Humans are unable to get metabolic energy from cellulose. Why? Well, cellulose, which you know, is a carbohydrate, but it's really hard for us to digest. We don't have the right enzymes to do it. So C. Number 43, DNA polymerase requires what? DNA polymerase requires an RNA primer, E, for it to function. Number 44, the central dogma of biology is stated as DNA to RNA to protein, A. 45, the allelic makeup of an individual is referred to as, well, that's their genotype. That's all the genes found in a particular organism. If it was their chromosomes, what is their chromosomes? That's a karyotype, but all their genes, that is a genotype. Number 46, 22% of the DNA from an organism is C. So what would the percentages of the others be? Well, figure out G and C need to match, so they should both be 22. And then you'll have to figure out what the proportions of the other 2 are. And the best answer here is C. Number 47, what would happen if you put a 10 molar solution of glucose into an artificial cell and place it into a beaker of 5 molar glucose? What would you expect to happen? Glucose can't cross the membrane. Well, you have to remember, then water will cross the membrane in order to balance out those concentrations. So water will move into the cell. 48, the process in which RNA polymerase assembles an mRNA molecule. What is that called? Number 48, that's C, transcription. 49, which of the following yields the most NADH per molecule of glucose? I believe that will be the Krebs cycle. Actually, I'd have to look at, I have to make sure I have that memorized correctly, but I believe it's B. You can check your own notes, double check that one. That's number 50. Complete the following analogy. Matrix is to mitochondria as blank as to chloroplast. So where do the hydrogen ions flow through ATP synthase? They flow through ATP synthase into the matrix in the one case and into the stroma in the case of the mitochondria. So the best answer there is B, stroma. 51. In eukaryotes, glycolysis reactions take place in the what? B, cytoplasm of the cell. 52. How many electrons does carbon have in its outer shell? The best answer there, 4. 53. Which of the genotypes below show only a homozygous condition? Well, that would be this one and this one. So the answer is 2 above, E. Why do phospholipids work well in a membrane component? The best answer there, they have both polar and nonpolar parts. So they make a double membrane, B. What disadvantage might a eukaryote species that reproduces only by mitosis experience? Why would that be a problem? Why would that be a disadvantage? The best answer here is going to be D. a lack of genetic diversity in offspring. So you won't have that diversity needed for evolution by natural selection to take place. All right. So I hope that was helpful, and good luck studying for your final exam, and I'll see you in class very soon.